Hello students, in this part we will discuss on sex determination. Sex determination. How is the sex determined in different organisms as well as a human being? The determination of sex is dependent on chromosomes. The number of chromosomes. As we have discussed already, the chromosomes, there are two types, X chromosome and Y chromosome. X chromosome, we know that chromosomes is basically two types, X body or X chromosomes, you can say that, and Y chromosomes. X chromosome or X body is proposed by H. Henke in 1902. X body was discovered by Henke in 1902 and Y chromosome was discovered by Nettie Stephen in 1905. So that sex determination can take place with the help of chromosomes. In different organisms there are different methods by which we can determine the sex. The sex determination is dependent on the different two methods. What is male heterogamity and second one is female heterogamity. Female heterogamity. Male heterogamity is basically two types, XX and XY, XX and X0 or X0. Male heterogamity, it means the pair of gametes are if different. Both pairs of gametes means such like as X0 and XY, X and Y, these are different. So these are the males. Males gametes जो होते हैं पेयर में हो different है उसको gamete hetero, and these are the male heterogamety XX and XY. XX and XY, this is present in the sex determination can take place this type of chromosomes in human being and Drosophila melanogaster. Human being and Drosophila melanogaster. You can say that fruit fly. In Drosophila or human being, the female contain similar type of gametes. Means this is the female and this one is male. Crossing process where we can represent by this crossing process of these homogamety of female with heterogamety of male can represent the sex in the newborn baby. We can use Punnett scale. This is the female and this one is a male. Female is XX and male is X and Y. When male X sperm fuses with X ovum, it produce XX, such like as XY, XX and X. Clear? So these are the cases of female. This one also female, and this one is male. You can say write this one male. So there is possibility of two males and two female. This sex determination can take place in human being as well as Drosophila melanogaster. In sex determination, Drosophila melanogaster is also used in another technique, but mostly in this technique it will be used. And second one is XX and X0. It means a female contains similar types of gametes. This is the female. And male contain one lesser means X0. It means number of one chromosome is less. this is the male. And this type of sex determination can take place in grasshopper. In grasshopper, a female consists 24 number of chromosomes, while in male it is contain only 23 number of chromosomes, means one chromosome is less in male than in female. The sex determination can take place by the Punnett scale, just like this one. A male consists X and naught, and female is X and X. X and X, XX, and then X naught, same as it is XX and X naught. So these are the females, and these are the males. Grasshopper contain one number of chromosome less than the female. Male grasshopper contain one number of chromosome less than the female. This is the male heterogametic. It means in these organisms, the male contain different types of gametes. And second one is female heterogameity, means the female contain different gametes. Different gametes means a pair of gametes which contain, those are different. Female heterogameity is also two types. We can also represent XX and XY same manner. But how we can differentiate between the female and male heterogameity? We can represent it another word, another letter, such like as ZZ and ZW. One is. And next one is ZZ and Z naught. Z naught, it means number of one chromosome less. ZZ and ZW, the determination of this type of female heterogameity may be present in birds, few fishes, कुछ मछलियों में भी होती है, and butterfly. But a few butterfly may show this type of female heterogameity sex determination. ZZ and ZW, the sex determination can be represented by the female heterogameity present in birds, in few cases of fishes and butterfly. It means a female contain different types of gametes. This is the female. Female contain different gametes and male contain homogametes. This is the male. Such like this one male and this one is female. We can again represent it by the Punnett scale. Male is ZZ, means homogametes, and female is ZW. So while we crossing, we can obtain such like this one. So if these are similar, this is the male. In birds, homogameity present in males and heterogameity present in females. These are the females. Clear? In few, butterflies and moth can represent this type of sex determination. Few butterflies, mostly butterflies show sex determination by the ZZ and ZW. But a few butterflies may contain in female one less chromosome than the male. And moth. This is the male and this one is female. One chromosome is less in female heterogameity in few species of butterfly as well as in moth. While we express in Punnett scale, we can explain it ZZ male and Z naught is female. While crossing ZZ, ZZ and Z naught, this one is also Z. This is male, male, and this one is female and female. Clear? So this is the technique how we can determine the sex determination in different organisms. We have already discussed Drosophila melanogaster. We can also obtain the sex determinant visual-like structure, and again counting the number of chromosomes. Counting the number of chromosomes. Fruit fly. in fruit flies we can also obtain or we can determine the sex by the number of counting the chromosomes. We know that there are two types of chromosomes: sex chromosome and autosomes. Sex chromosome that represented by XX and XY in the Drosophila, and autosomes means they make the body structures. So in fruit fly we can calculate the number of X chromosomes as well as autosomes. If the ratio of X chromosome and autosome is 0.5, it means such like as X is 1 and autosome is 2, then 1/2 is 0.5. The fruit fly may be male. How we can calculate? We can calculate the X chromosome and autosomes in the fruit flies. If the ratio of X and autosomes is 0.5, the production of the fruit fly is male. And if the ratio of X and autosomes is 1, then the fruit fly will be female. Clear? But one more possibility, if the ratio of X and A chromosome between 0.5 to 1, 0.5 to 1, 0.5 is male, 1 is female. If the ratio of 0.5 to 1, between the 0.5 to 1, then this type of fruit fly may contain female as well as male features, and this is called gynandromorphs. If the ratio of X and autosomes may be between 0.5 to 1, this type of fruit fly contain male and female features, and this male or female feature is known as gynandromorphs. These gynandromorphs may be three different types: anterior post means anterior portion contain, frontal portion contain male and posterior portion contain female, maybe vice versa. And bilateral symmetry. Bilateral means half portion act as male and half portion will be shown as female. That is the bilateral. Gynandromorphs is three types. Anterior post means anterior post contain the male and posterior female, male and anterior female. That is the difference. And second is bilateral. It means one half portion of the fruit fly may show as male part and other one is female. And third one is sex pairs. Sex pairs. It means the body of fruit flies is maximum females, but there are some pouches of male can be present. Tissue, cells and body all contain female. Sex pairs means the fruit fly body, tissue and cell all are female, but there are some patches of male present. That is called sex pairs. We can calculate it by the counting the X and Y autosomes. One more possibility, if X autosomes and sex chromosome ratio is less than 0.5, means 5.5 है तो मेल था, 0.5 से less है, then that is called metamales, or you can say that super male. If the X and autosomes ratio is more than 1, one था तो कितना था female, more than 1 है, that is meta female or super female. That is meta female or super female. So we can determine the sex in Drosophila melanogaster by the X and XY chromosome as well as the counting the X and autosomes. Clear? And in few Reptilia, the sex determination can take place with the help of temperature, means incubation time period. The body heat provided by the organism to its egg. Incubation time period is also represent the sex of the new offerings. What is incubation time period? The body heat which is provided by the animals to its egg, that is called incubation time period. This can be seen in some reptilia, in crocodile and lizard. If the body heat provided by the organism to the egg is less, then the progeny will be produced in female. If the incubation time is less in crocodile and lizard, the female progeny will be produced. Or if it is higher, then male progeny will be produced. But in turtle, this is the opposite. In turtle, it is opposite. Higher body heat, higher incubation time period may produce the female, and less incubation time period may produce a male. This is the another one technique by which we can determine the sex. One more type in bees. In bees, there is different types of sex determination process can take place. In bees, there are two types of eggs: fertilized egg and non-unfertilized egg. Fertilized egg. While fertilization can take place, there will some eggs may be fertilized and some remains unfertilized. Fertilized egg is diploid and unfertilized eggs are haploid. Clear? Unfertilized eggs due to another techniques, parthenogenesis. Parthenogenesis. By the process of parthenogenesis, unfertilized egg give birth to the new offspring, means male bees, and those are called drone. So we can say that the unfertilized egg give birth to the male bees, or you can say that drone. The reproduction or development of any organism or male bee without fertilization or fertilized egg, that is called parthenogenesis. So by the process of parthenogenesis, unfertilized egg change into the male bees, and those are called drones. If the egg is fertilized, egg fertilized होते हैं तो ये दो प्रकार से. On the basis of nutrition, they may produce different types of bees, and these bees are females. On the basis of nutrition, on the basis of nutrition. अब न्यूट्रिशन के बेस पे यहाँ जो बीज प्रोड्यूस होती है, पहली बात तो दो फर्टिलाइज एग से बीज जो प्रोड्यूस होंगे फीमेल होंगे, और जो न्यूट्रिशन इनको प्रोवाइड किया जाता है न्यूट्रिशन के बेस पे दो प्रकार की रॉयल जेली. यदि इनको रॉयल जेली के रूप में न्यूट्रिशन दिया जाता है, दैट में प्रोड्यूस द क्वीन बी, बट इट इज ए कंडीशनल. Queen bee, royal jelly will be produced, or queen bee will be produced at the time if the hives cannot contain queen, may be died or fly from one hive to another, and the hive will remain without queen. So royal jelly are produced to provide to the fertilized egg, and this may produce a queen bee. And second one is bee bread. If another nutrition can be provided to the bee bread, then these bees are also, queen is also female, it only function is to laying eggs, and male is only function to fertilize the female queen bee. And if the nutrition will be provided to the bee bread, then that may produce sterile female bees, and these are also known as worker bees. So we have discussed how the sex determination can take place in different organisms. These are the incubation time period, as well as what is parthenogenesis. Development of any organism without fertilization, that is called parthenogenesis. Drone bee is developed from the unfertilized egg. Haploid and diploid fertilized egg may produce two different types of bees. Both are female. One is queen bee. It depends on the condition and provide to the nutrition. If the royal jelly will provide to the fertilized egg, then it may produce queen bee. Or if bee bread nutrition can be provided to the fertilized egg, and it may produce a worker bee, and these worker bees are sire. Clear? This is the sex determination can take place in animals.